timing is the most important, because back in 2006, I will just shut up, stop, no perfusion. 2008, even at the meeting, no perfusion in this company. We will never ever run. I still remember this sentence. We will never ever run perfusion in this company. And just timing, right time, same ID, right people in the meeting, just say the same, and then it was just accepted. And since now it's a platform in this company for many products. It's just timing, learning. Even if you have data, sometimes it's not enough.
Welcome back to the Smart Biotech Scientist. I'm your host, David Brühlmann, and today we're diving deep into continuous processing with Jarno Robin, who has spent over 20 years mastering upstream continuous processes at industry giants like Novo Nordisk, Sanofi, and Leo Pharma. Now at Sani Membranes, he's leveraging the potential of Vibro Membrane Filtration technology, a revolutionary membrane filtration that eliminates pressure loss and reduces fouling. Let's explore his incredible journey together. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brühlmann, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. Welcome, Jarno, to the Smart Biotech Scientist.
Thank you, David.
Yeah, great to have you on today.
Yeah, I'm very excited to have this interview together with you and to have a nice discussion.
Jarno, share something that you believe about bioprocess development that most people disagree with.
There's a couple of ones, actually. I've experienced myself this. The first one, I will say, it's when you make a process development, typically you start in the labs, get very nice results, everybody is happy. But I will say and recommend, it's good to have higher yield as possible. Maybe if you take a fed batch for antibodies, reaching 10, 15 grams per liter, perfect. But when you transfer to a large-scale pilot plant or production scales, if the yields go down to 3 grams per liter, that's a nightmare. So I always recommend to find the right compromise and maybe enjoy with 7 grams per liter in the labs, but then having a 7 grams in the facility. So that's very important that you find this compromise, that you have something which is robust at large scale, because it's at large scale you will earn money at the end of the day. So that's one of the most important one, I would say. And then the other one, it's when you make a process development, I have seen before that a lot of people always run a control. So if you change the process parameters, people want to have a control to compare. It's good to have a control, but I will say not each time. Because at the end of the day, you will generate a lot of data on your control and not on your new process. So I will say at the beginning, when you make your DOE, design of experiment, try without a control to see, because you want to see higher increase in the yield. And then only at the end, then you can make a control, because then you have developed and spend your time right on developing your process to something better than to develop your control with plenty of data on your control. It's not needed. Probably not all people will agree on that, but that's the way I've done it, and that's the way I've tried to share my experience to young scientists.
Yeah, Smart Biotech Scientists, we would love to hear from you. Should you do control or not, or in what situation? So looking forward to hearing from you. Now, before we dive into our conversation on continuous processing and filtration and a lot more, Jarno, tell us about your story, because you had this incredible 20 plus year journey through some of the biotech biggest names. So what was that one moment early in your career when you thought, well, this is it, continuous processing is the future? And what were some interesting pit stops along the way?
It started already from almost the beginning, because I did my industrial PhD with DSM Biologics, and they use chemostats So it's a kind of continuous processing. And it was very interesting because you could play with the setup and you get a reach a steady state. And then you can change a process parameter, reach a second steady state. And then you can come back again, reach a steady state. So you could really play running the same experiment and play with a different process parameter. So already there, say, okay, that's pretty smart because if you run a batch or fed batch, then you run 2 or 3 days and then switch off and then clean and restart. And then you never know if there was some difference happening, and then you will never maybe recover the same data, same results. So already there during the PhD, I could see the benefits of continuous processing. Then when I started to a small company here in Denmark, actually the platform for the process in upstream was continuous. It was perfusion with mammalian cells. So it was just a natural way, and to move forward with continuous processing, could see the benefits of the perfusion process when you reach very high cell density, high yield, plateau. So basically, when you integrate under the curve, you have highest yield you can get there. At that time, it was early 2000, so there was some issues for scaling up with the cell rejection device. The ATF was not there. TFF was not well known for mammalian cells because of peristaltic pumps, not a good combination at that time. So there was some limitation. It was ultrasonic system we used, so limited to 500 liter or something. So at that time it was, but I could really see the benefits. And then I moved to Novo Nordisk here in Denmark. And at that time, Novo, this company was running a batch. And it was a batch culture. And I will say it was in 2006 where we were using a very bad cell line for antibodies production. And I could I run in my interest hours perfusion just to see the results. And every day I got the same yield than what I got after 14 days in the fed batch. So basically the yield was 14 times higher. Then you can run and run and run. And I remember when I showed the results to the project manager, she just killed me completely down. She said, you waste your time. It was not needed. It's just because the batch culture, not the perfusion. Even the data was very clear, 14 times higher yield. Then you go back to fed batch. But I was already convinced about perfusion. The funny thing is that 2 years later, 2008, there was a need for a project on phase 3 to get the yield much higher, and it was a repeated batch process. So basically you run 3 days, harvest 90% of your bioreactor, the fresh medium, 3 days, very robust, very good. But the yield was extremely low. And I remember I had a look at that time to the ATF there, and we make some simulation in the lab, and we got 6 times more in yield compared to the current process. And I say, okay, so I make some slides, and my manager say, nice slide, but just keep them and let me tell you when you can show them. Then we went to a meeting, and the funny thing, it's a project manager was the same project manager than for 2 years ago. And we had the senior vice president in the room in the meeting. So we start trying to optimize the current process, and maybe we could get 10-15% more. It was not enough. And then I do my manager and just make the sign with the head, move forward, Jarno. And when I start my presentation with the first slide, I pronounce the word perfusion. Then the project manager just react and just say, no, we never ever run perfusion in this company. And I remember as a senior vice president looking to her and say, if you don't listen to him, he didn't start. So why you want him to stop? And if we don't listen to him, then we just close your project. So what do you want? So I had the permission to move forward and make the slides. And I could see on the project manager, she was just very angry. But senior vice president just loved the presentation because you can say we make simulations 6 times higher yield, the first dollar. And then second question, he said, which technology do you want to use? Is it scalable? I say, yeah, the ATF looks that you can go in the lab, but also large scales. So then second dollar and say, but how much does it cost? I say, it costs something around 150,000 euros per unit in a large scale. And the guy knew how much billion he could earn the $dollar on a base, and they say, okay, fine. And the project manager, it took her 4 years to accept that we make the change. We had 6 months. It was in December, and in July, we had to make the first of phase 3 batch. So 6 months to implement a new process, a new technology, both in the lab and in the facility. But then after that, when you run in the large scale 6 months later and you get the 6 times yield increase and the product quality was just good, then it's a funny story, but that's the way we move forward to the ATF or perfusion processes.
Well, what a fascinating story. I mean, that's amazing. It shows us actually well how conservative the industry is where we are working in, because the numbers are clear, 6 times higher yield. And as we know today, a continuous process has a lot of advantages, but still that was not sufficient or took that long to convince your management. That's crazy, isn't it?
I mean, I learned from that time, and that's also my message, take-home message for people listening, but also as a young scientist. timing is the most important, because back in 2006, I will just shut up, stop, no perfusion. 2008, even at the meeting, no perfusion in this company. We will never ever run. I still remember this sentence. We will never ever run perfusion in this company. And just timing. Right time, same ID, right people in the meeting, just say the same, and then it was just accepted. And since now it's a platform in this company for many products. Just timing, learning. Even if you have data, sometimes it's not enough.
Yeah, that's excellent advice, smart biotech scientist. Timing is everything. Timing is so important. Since, Jarno, you have implemented a lot of perfusion processes, you were one of the pioneers very early on. You believed in it. You stuck with it despite the pushback. Tell us a bit what perfusion looks like today and what are the technologies we're using? What is the state of the art today? And what are the advantages and still the challenges we're facing?
Oh, yeah. I mean, right now, it's still ATF and TFF for cell retention device in the upstream, mainly. All this very old-fashioned acoustic system. I know some people still use them, but it's the biggest volume you can run. It's 500 liters, so it's out. And then centrifuge, you have a couple of companies running with single-use centrifuge out there. But then never breaks through just because of the technology. People who tried centrifuge in early 2000, I can tell you, then the bag just break down and you have the leakage, everything was on the floor. I think there was this, remember how it was. And also when you want to go to large scale, then you have a huge system in your facility. So the footprint is just enormous compared to the ATF or TFF. And footprint is also very important in a facility. You want something very small, because the footprint is expensive in facilities. So today, most of perfusion are using ATF or TFF.
So if I may interrupt you real quick, just tell us what is ATF and what is TFF for those who are not familiar.
Okay, ATF, alternating tangential filtration, and TFF is tangential filtration. I mean, TFF was the first technology, I would say, where you have a hollow fiber cartridge, and then you have a pump circulating inside the hollow fiber, and you harvest through the fiber. So the harvest is also cell-free. That's also an advantage of the ATF and TFF with our hollow fibers. The harvest is cell-free. It's not the case with acoustic system or centrifuge. You need a filtration step after that to remove the GMO material. So that's also the beauty of the ATF and TFF. And then came the ATF, alternating tangential filtration from Repligen, where you have this diaphragm on the bottom with compressed air and vacuum. So you get the diaphragm to move up and down inside the hollow fiber. So that's why it's called alternating, because it's going in both ways inside. So you have only one connection to your bioreactor. It's much more simple and work pretty well, we say. But both system, it's hollow fibers based. So due to the design, how it's made, you have a lot of pressure loss in the system, and that's just physics. So basically, you cannot change the physics. So you have to work with it. And I know a lot of people are working, developing fancy ways of running TFF or TF to reduce the fouling of the hollow fiber. Because that's one of the major issues with TFF or TF for upstream running perfusion. It's the fouling of the membrane. So already after 7-10 days, when you reach your high cell densities, then you can see that the product concentration in the harvest is going down. So basically, you're running more and more ultrafiltration, because then you up-concentrate your product in the bioreactor. And typically on perfusion today, you have a bleed stream to remove some cells to keep a nice viability, a healthy cell in the bioreactor. But at the end of the day, your main harvest is not what is going through your ATF or TFF, but it's in your bleed stream. So if you make the mass balance, actually, in some cases, half of your product is throughout in the bleed. So that's the limitation. And I know also in my position today that people are looking how to use the bleed to recover the product. And just by doing so, you can double the yield. So even if you run with this fouling issue with the ATF or TFF, as we said before, you can get 10 times higher yield. Still, if you treat the bleed, you can double up your yield. So a lot of people are focusing now on the bleed stream, which is actually can become your main harvest line.
As a scientist, you can read every peer-reviewed paper and listen to every podcast, but there is nothing like being in the room with industry peers, where you get new scientific insights, deals get made, and problems get solved. That's exactly why you need to be at the Advanced Therapies Europe 2025, which will take place in Barcelona from September 2nd to the 4th. Connect with 500 plus cell and gene therapy leaders solving the same challenges you face. I'm talking live case studies on cost reduction strategies, direct networking with companies actually implementing these solutions, regulatory insights on distributed manufacturing models, and exclusive collaboration opportunities that you won't find anywhere else. As a Smart Biotech Scientist listener, you will get 20% off with this code SMARTPODCAST20. That's SMARTPODCAST20. Look, your competition will be there. The question is, will you? Register now. You will find the link in the show notes. What I'm hearing, Jarno, one of the biggest challenges with ATF or even with TFF is the foulings. And what are some other limitations? Is the scale-up still an issue, or where are we there, or the robustness of these systems?
Scale up, it's quite okay. I mean, I am not the issue. You need to understand what happened in a large scale with the mixing, the gas mixing. So you need to take this in consideration in your lab and try to mimic this in your lab. And sometimes you need to accept that what you do in the lab, it's not something you will do in the large scale, but it just make a nice model. So as long as you can reproduce in the lab what you have in the large scale, that's the most important. It doesn't matter the way you are doing it. For example, for one product, when I run a microsparger in lab scale, the product was completely just destroyed. But when I switch in the lab scale with a ring sparger, then the product was just perfect. And then when you scale up, when we run the first 100 liter, we just say, okay, microsparger or ring sparger? We try with ring sparger. But after one day, as a technician came to me and said, we empty the whole bottle of oxygen, and it cost 4,000 euros per bottle per day. I say, okay, now we'll switch to micro sparger, and the product quality was good. So basically, we had a setup for the lab, working fine. But for large scale, you move to another setup. Sometimes you need to be open-minded to try things and get it to work. But the technology also, TFF, ATF, are pretty robust, I would say. Also, it's quite okay. That's why people are using it. For the ATF, you can get it as a single-use system, gamma-irradiated system, or as a stainless steel, and you can autoclave it. Then you can face some challenges with the autoclave. We spent 6 months to get a program to autoclave the ATF. And basically, when I had to transfer this process to another site, the guys say, oh, we can make autoclave program. I say, no. You just get it. You have the recipe here. You just copy-paste and don't change it. We spent 6 months on that, millions of euros spent, because each time we just destroyed the whole fiber. It was leaking, and then infections. I mean, it was just a nightmare. It went to the very top of the company because we were stopped in the middle of phase 3 production because of this. So autoclave can be a huge challenge for those technologies. So you need to be very aware. And I know that if you run single use, then the price is pretty high. So you really need to think about your business case, what you want to do, and the facility you have and the knowledge you have inside.
And despite all these challenges, so you mentioned the pressure loss or the fouling, also the high costs or the autoclaving, TFF and ATF have become the gold standard in upstream, but also in downstream. But despite of that, you're now working on an exciting new technology, a new way to filter. And tell us a bit more why you started this development and what are you trying to solve and how is your technology different?
I had this background, as we just discussed, about running perfusion in the upstream, and I know that TFF is pretty well used in the downstream for UF ultrafiltration applications. But when I got contact with this company, Sani Membranes here in Denmark, through a friend who was in contact with the CEO, and when he explained me about the technology, I say, wow, that's lovely. You can use this technology for both upstream and downstream. And design itself, it's not hollow fiber based. It's membrane based. So you have already on the market TFF running with cassettes with membrane. But here the design is completely different. Typically in the TFF systems, you are using a pump to generate turbulence on the membrane and the hollow fiber. Since you have foulings issue, it just shows that the turbulence are not enough. And use a huge pump, so a lot of energy consumptions. And sometimes all pumps just generate heating. So some customer need a cooling unit after the pump to cool down, otherwise the product will be destroyed. So it's a lot of energy and still not efficient. Here's this technology, it's membrane filtration, but it's vibrating. And basically the vibration creates turbulence right at the surface of the membrane where it's needed. So you still need a pump to get things circulating inside the cartridge, but it's not for generating turbulence because the vibration will do it. So it's a very simple way. I think PAL did it for 20 years ago, but they just stopped because they couldn't scale it up. There was a lot of issues. But here's this technology. You can work in the lab and you can scale it up to 80 square meters, so a huge facility. And behind me, I have a picture where for algae, you have 3 or 4 of this 80 square meter in a row. So you have almost no limits for scaling up. And then when you go deeper in the technology, the way it's designed, it's 70% more open compared to any other system. So that means you can run up to a very high viscosities, Greek yogurt, up to 2000 CP. And for me, I was just, wow. Because one of those limits of the TFF and ATF, when you run perfusion in the upstream, when you reach above 100 million, 150 million cells per milliliter, then you start to not only have fouling of the membrane, but blocking of the hollow fibers. And then it will just block and stop. Here, since it's much more open, no pressure loss, uniform TMP, you use the whole surface area. And the beauty of this viscosity, you can run at much higher cell densities. So the potential for me, I would just higher cell density, higher yield. And then probably you will move the limitation back to the bioreactor with all the mixing, gassing and everything, and not cell device retention anymore. So I just see a lot of potential, and I've been 2 years in this company, and I see still a lot of potential, but conservative. This industry in pharma, even they have good data and all the issue with the ATF or TFF, they are still happy they have it. So you need to convince, and again, timing is very important to find ambassador inside companies that are willing to test. So that's the beauty of this technology, because it just opens some new ways of running. You can run also on the downstream, just change the cutoff of the membrane, where you can work from a 1 kilodalton to 800 kilodalton for ultrafiltration. So a large pool of membrane, 30 to 40 different membranes, different material. So you can really play with different parameters. So that's also flexibility in this technology. The other part is the energy consumption, sustainability. Maybe we'll discuss that a bit later, but it's using almost no energy. And in the pharma industry, it's more and more important to take it in account.
The part is that because you have a low and uniform membrane pressure, or why do you use less energy? Because you also have to vibrate the membrane, right?
Yeah, but that's the beauty. First, you start with a smaller pump, so less energy consumption. You don't need cooling units. So you remove cooling units if you need it before, because it's a small pump, so you don't generate that much energy anymore or heating. The pump uses much less energy, and the vibration is using almost no energy. For the biggest system, the 80 square meter, it's only using 3-4 kilowatt per hour. So it's nothing. If you compare with the ATF, where you need compressed air and vacuum, you need 2 compressors. One for creating vacuum, the other one for compressed air. And typically in a GMP facility, you have a double up, you have a backup of both. So you have 2 compressors running continuously, and that's sucking a lot of energy. That's crazy. With this system, you use 90% less energy.
Yeah, 90%, that's massive. Wow.
This technology is also used in other fields like algae, and they are very taking care about energy consumption. And that's true. I mean, this technology is using almost no energy compared to any other technology.
What about the shear stress? I assume that that's low too, because you have a low transmembrane pressure, right?
But that's a lot of questions. This question is, what about the vibration? Do you destroy the product or cells or whatever? And basically, today we have quite a lot of data on the fed batch harvest clarifications, where typically you have a centrifuge and depth filtration. So here you can remove both steps directly on the Vibro system. After 4 or 5 hours running the process through the Vibro system, the cells are intact. No cell debris, so viability is still the same. Cell count is still the same, so it's unchanged. So that's why I have a huge expectation for perfusion to see if you run continuously 24 hours, 7 days a day, 100 days, how does this look like? But there is a lot of parameters with the pumps and everything. So it's going slowly in this direction. But for the harvest clarifications, results were very good. We just got our first articles published with messenger RNA, where basically the performance are 3 times higher than the classical TFF today from competitors. And when they looked to the pro quality of the messenger RNA, because you have the structure and everything, it's intact. It's a classical question, a vibration. Can it destroy the product or the cells? So far, what we can see, no.
That wraps up part 1 of our deep dive with Jarno Robin. We've explored his career journey and the compelling benefits of continuous processing. In part 2, we'll tackle the technical challenges and discover the game-changing potential of Vibro Membrane Filtration technology. Do you have process development questions? Well, book a free consultation with me at brühlmannconsulting.com. I'm here to help you navigate these complex decisions. And thank you so much for tuning in, and I'll see you in part 2. All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.